In this module, we shall learn about the reactions catalyzed by zinc metalloenzymes, carboxypeptidase A and human carbonic anhydrase 2, and the catalytic mechanisms of these reactions. The role of different amino acids in the active site pocket of the enzymes will also be discussed. In this module, we shall learn about the reactions catalyzed by zinc metalloenzymes, carboxypeptidase A and human carbonic anhydrase 2, and the catalytic mechanisms of these reactions. The role of different amino acids in the active site pocket of the enzymes will also be discussed.
Dear students, you are already aware that zinc is, although a trace element, but it is an essential constituent in blood and several tissues. You are familiar that an adult has about 1.5 to 3.0 grams of zinc, and the largest amounts are present in liver and bone. There is evidence that zinc concentrations in blood and several tissues vary considerably in response to many stimuli. Zinc appears to be critical in many functions. Human zinc deficiency is inherited in infants and is termed as acrodermatitis enteropathica and is characterized by behavioral disturbances, diarrhea, hair loss, and severe perifacial skin rash, all of which respond with remarkable promptness of administration. Similar syndromes have now been reported many times with penicillamine treatment of other disorders, presumably due to the chelation of the zinc, as well as during total parenteral nutrition when zinc was not added to the nutritional solutions for even as short a time as 2 weeks. A more chronic dietary deficiency of zinc includes dwarfism, hypogonadism, and sexual immaturity, the latter devised with zinc therapy. There is much evidence for marginal dietary deficiencies in humans. The effects includes decreased activity of taste, impotence, delayed bound healings, hypogonadism and oligospermia, pure development and possible fetal wasting and teratogenesis. Dietary source range in zinc concentration from 1400 microgram per gram to 2 gram per gram or less in fresh food and vegetables. Viability of zinc is essentially high from animal tissues and is low from animal and from grains. The latter effect is apparently due to the binding of phytic acids and fiber. Zinc appears to be critical in many functions as it is an integral constituent of various enzymes which are present as proteins in biological systems. This and the next module shall focus on zinc enzymes. Some common zinc proteins are carboxypeptidase, carbonic anhydrase, alkaline phosphates, alkaline phosphatase, dipeptidase, insulin, etc. In this module, we will understand how zinc 2 ions acts as a good Lewis acid to catalyze condensation and hydrolysis reactions at physiological pH. We shall study in detail about the structure, active site, reactions of the enzymes, carboxypeptidase A and carbonic anhydrase 2. These two zinc proteins play a significant role in biological system.
About 300 different zinc proteins are known. The zinc in these proteins has either a catalytic role in enzymes as Lewis acid, regulatory or a structural role. The table in this slide gives the names of some zinc-containing proteins along with their functions. In this module, we'll be studying the zinc proteins carboxypeptidase A and carbonic anhydrase in detail. Dye-positive zinc ion is a good Lewis acid and when present in metalloenzymes can catalyze condensation and hydrolysis reactions at physiological pH by polarizing the substrates. Some of the enzymes catalyzing such reactions are shown.
Carboxypeptidase A, which is abbreviated as CPDA. Carboxypeptidase A is one of the most studied peptide underlying enzymes. In 1929, Waldschmidt, Leeds and Poor isolated carboxypeptidase A for the first time. It was the first metalloprotease and second zinc enzyme to be identified. There are two kinds of carboxypeptidases. Enzymes of one class are not metalloenzymes. Yeast carboxypeptidase C belongs to this class. The second class of carboxypeptidases are metalloenzymes. Carboxypeptidase A is an example of this class of enzyme. It is an exopeptidase A, that is one which cleaves only terminal peptide bonds, that hydrolyzes a peptide bond at carboxyl terminal, that is C terminal, end of the peptide or the protein. A related protein carboxypeptidase B catalyzes the hydrolysis of basic amino acids lysine, arginine or methionine from C terminal position of the polypeptidase. Hydrolysis by carboxypeptidase A occurs only if the C terminal residue has S configuration and its carboxyl group is free. Peptides possessing any C terminal residue except proline will be hydrolyzed and an aromatic amino acid side chain at the carbon terminal residue is favored in the substrate.
Carboxypeptidase A is an exopeptidase, that is, a peptidase which cleaves only terminal peptide bonds. It hydrolyzes a peptide bond at carboxyl terminal or C terminal end of a peptide or protein. It has a single protein chain of 307 amino acids with one divalent zinc present. In the absence of the enzyme, the hydrolysis reaction is slow, with rate constants of the order of 10 to the power minus 11 second inverse. However, if the reaction is catalyzed by carboxypeptidase A, the rate constants can attain values of 10 to the power 4 second inverse. Carboxypeptidase A is initially produced in an inactive form called procarboxypeptidase A in the pancreatic juice of animals. This inactive form is converted to the active CPDA by the enzyme entropeptidase. As we can see in this figure, the active site of carboxypeptidase A has one dipositive zinc ion with a coordination number of 5. This zinc is coordinated to two imidazole side chains from histidine 69 and histidine 196 to the bidentate carboxylate group of glutamate 72 and one water molecule, giving it a distorted pentacoordinate geometry. Many acidic and basic amino acids, namely asparagine 144, arginine 145, tyrosine 248, arginine 127, and glutamate 270 are also present in the active site pocket and are important for the activity of the enzyme. The active site cavity also has a hydrophobic pocket that accommodates the non-polar R' of the C-terminal amino acid of the substrate undergoing hydrolysis. As we have just seen that in the active site of carboxypeptidase A, amino acid glutamate 270 is present. The water molecule coordinated to dipositive zinc forms a hydrogen bond to the carboxyl group of glutamate 270. This interaction converts the bound water to nucleophilic hydroxide, which then attacks the carbonyl carbon of the cisyl peptide bond, as will be shown in the next slide. Now that you are familiar with the active site of carboxypeptidase A, we will try to get an insight into its catalytic mechanism. The first step in the mechanism is positioning of the substrate on the enzyme, as shown in B. The enzyme interacts with the substrate with hydrogen bonds and van der Waals forces. Arginine 145 and asparagine 144 form hydrogen bonds with terminal carboxylate group. Tyrosine 248 with the penultimate peptide NH and arginine 127 with carbonyl oxygen of the terminal peptide bond. There is van der Waals attraction between the hydrophobic pocket and hydrophobic R' of C-terminal amino acid if the terminal group is aromatic. The interaction of arginine 127 with the carbonyl of the terminal peptide bond increases the Lewis acidity of the carbonyl group, making it more susceptible for nucleophilic adhesion. Then the water molecule bound to zinc performs a nucleophilic attack on the carbonyl carbon of the C-terminal peptide bond, with the simultaneous transfer of a proton to glutamate 270. At this point, glutamate 72 bound to zinc ion becomes monodentate from bidentate, maintaining the 5 coordination of zinc ion. The tetrahedral intermediate formed by the nucleophilic attack is shown in C. The protonated glutamate 270 now transfers its proton to the amide group of the terminal peptide bond. This causes the collapse of the tetrahedral intermediate, resulting in the formation of the free carboxyl terminal amino acid and the shortened peptide chain with the terminal carboxylate group bound to zinc. A water molecule adds to the zinc ion. Finally, the cleaved peptide leaves and zinc ion gets back to its original pentacoordination.
Carbonic anhydrase. After studying about the carboxypeptidase A zinc protein, let's know about another very important zinc protein, that is the carbonic anhydrase, which is also known as human carbonic anhydrase. The first zinc metalloenzymes, carbonic anhydrase, was discovered in 1940 by Kellin and Mann. It is a lyase that catalyzes the reversible hydration of the carbon dioxide to form bicarbonate ion. In the absence of the enzyme, this reaction of conversion of hydration of the carbon dioxide to form bicarbonate occurs relatively slowly at physiological pH and the presence of carbonic anhydrate increases the rate up to a million-fold. In vitro, it catalyzes several other reactions also, such as the hydrolysis of the esters and the hydration of the aldehydes. Carbonic anhydrases are found in all kingdom of life, occurring in animals, plants, and several bacteria. They play an essential role in biological processes like respiration, photosynthesis, and in maintaining pH. There are several forms of carbonic anhydrates. All the forms are single chain polypeptides with molar mass about 30,000 Da and have one zinc ion per molecule. High activity forms have K catalytic activity of the rate of 10 raised to power 6 S inverse at 25 degrees centigrade and are labeled 2. Low activity forms and the very low activity forms have K catalytic activity 10 raised to power 5 S inverse and 10 raised to power 3 S inverse and are labeled as 1 and 3 respectively. The best studied carbonic anhydrase is carbonic anhydrate 2 from red blood cells.
The second enzyme we will study is carbonic anhydrase. It is the first zinc metalloenzyme discovered in 1940 by Keelan and Mann. It is a lyase that catalyzes the reversible hydration of carbon dioxide to form the bicarbonate ion. Carbonic anhydrases are found in all kingdoms of life, occurring in animals, plants, and several bacteria. They play an essential role in biological processes like respiration, photosynthesis, and in maintaining pH. There are several forms of carbonic anhydrases. The best studied carbonic anhydrase is Ca2 from red blood cells. As can be seen in the figure, human carbonic anhydrase 2 contains dipositive zinc ion at the active site. This zinc is coordinated to the imidazole nitrogen atoms of 3 histidine residues. Histidine 94, histidine 96 are ligated to zinc via their N-epsilon atoms, and histidine 119 is bound via N-del atom. The 3 histidine NH protons are all engaged in hydrogen bonding. Histidine 94, histidine 96, histidine 119 are involved in hydrogen bonding with glutamine 92, asparagine 244 and glutamate 117 respectively. The fourth coordination site of zinc is occupied by a water molecule to give a distorted tetrahedral geometry. The active site pocket also contains ordered network of water molecules and other amino acids like threonine 199, glutamate 106, histidine 64 that are important for hydrogen bonding, proton transfer, and for binding the CO2 substrate. Human carbonic anhydrase 2 has 3 histidine coordinated to zinc. Histidine being neutral and less basic than glutamate anion makes the zinc more acidic in human carbonic anhydrase 2 than in carboxypeptidase A. The binding of water to zinc reduces the pK of water from 14 down to 7. This leads to its deprotonation at neutral pH to form the strong hydroxide nucleophile. The dissociated proton is transferred indirectly to histidine 64 via water molecules. Histidine 64 shuttles protons from the active site to the buffer in the solution, shifting the equilibrium in the forward direction in favor of the formation of hydroxide ion. The enzyme then positions carbon dioxide for nucleophilic attack by the hydroxide. The nucleophilic attack by hydroxide on carbon dioxide produces a zinc-bound bicarbonate. Addition of water displaces the bicarbonate ion, completing the cycle. In this module, you have learned that carboxypeptidase A and human carbonic anhydrase 2 each have one dipositive zinc ion present at the active site of the enzyme. Carboxypeptidase A is a hydrolase that hydrolyzes the peptide bond at carboxyl terminal or C-terminal end of a peptide or protein. The catalytic mechanism involves nucleophilic attack by hydroxide on the carbonyl carbon of the C-terminal peptide bond. Carbonic anhydrase is a lyase that catalyzes the hydration of carbon dioxide to form bicarbonate ion and proton. The catalytic mechanism involves 4 steps: deprotonation, carboxylation, formation of a ring structure, and addition of water.
Let's now summarize what we have learned in this module. We have understand how zinc ions acts as a good Lewis acid to catalyze condensation and hydrolysis reactions at physiological pH. We have studied in detail about the structure, the active site, the reactions of the enzymes of carboxypeptidase A and carbonic anhydrase 2. These two zinc proteins play a significant role in biological systems. Carboxypeptidase A is a hydrolase that hydrolyzes the peptide bond at carboxyl terminal, that C terminal. End of the peptide are the proteins. Its active site contains one zinc ion, and this zinc ion is coordinated to imidazole nitrogens from two histidine residues, histidine 69 and histidine 196, two carboxylate oxygens of the glutamate 72 and a water molecule. Many acidic and basic amino acids, namely S-pyrazine 144, arginine 145, tyrosine 248, arginine 127 and glutamate 270 were also present in the active site. The catalytic mechanism involves nucleophilic attack by hydroxide on the carbonyl carbon of the carboxy terminal peptide bond. Nucleophilic, that is the nucleophilic addition reaction. Carbonic anhydrase is a lyase that catalyzes the hydration of the carbon dioxide to form bicarbonate ion and a proton. Its active site contains zinc 2 ions coordinated to the imidazole nitrogen atoms of the 3 histidine residue such as histidine 94, histidine 96, histidine 119 and one water molecule. An ordered network of water molecules and amino acids like threonine 199, glutamate 106, histidine 64 are also present in the active site pocket. The catalytic mechanism involves four-step deprotonation, carboxylation, formation of a ring structure, and addition of water molecules, resulting in the formation of bicarbonate.